Feldman Barrett, past president of APS, and it's my honor to introduce this presentation by Neil Burgess in recognition of his 2020 APS William James Fellow Award. The William James Fellow Award is APS's highest honor, recognizing members for their lifetime of significant intellectual contributions to the basic science of psychology. Neil is a professor of cognitive and computational neuroscience at University College London, where he studies various neural mechanisms supporting memory, with particular interest in the role of the hippocampus in spatial and episodic memory. But he also has an interest in the role of other brain regions and other forms of memory, such as phonological working memory. Congratulations, Neil, on behalf of myself and the entire APS Board of Directors.
Thank you, President Feldman Barrett, and I'd like to thank the APS for the William James Fellow Award. I feel very honoured to receive this award and to be associated in any way with William James and the previous recipients. I'd also like to thank all of my colleagues and collaborators over the years, especially Graham Hitch and John O'Keefe, who first introduced me to psychology as a naive physicist in the early 90s. And also to all of the talented colleagues and lab members I've had the pleasure to work with, and to UCL and my funders. I've always been interested in memory and in understanding how memory results from the firing of neurons in the brain via computational modelling. I'm also interested in spatial cognition, which allows a cross-species approach with similar tasks in rodents and humans. In this presentation, I'm going to consider the multiple ways in which we remember where things are and how they're implemented in the activity of neurons in and around the hippocampus, shown in yellow on the rat and human brains here, and in the parietal and retrosplenial cortices. At the end, I'll speculate a little about how this system might also organise conceptual knowledge. The spatial tasks allow for parametric manipulations, like this classic experiment from Wang and Simons. They ask subjects to view an array of objects on a circular table, and after a short delay, say which one had been moved. During the delay, the subject could move to a new viewpoint, or the table could rotate by the same amount, or both could happen so that the view of the objects was the same at encoding and at test. They found there was a benefit of consistency between the views at encoding and test, compared to when the table rotated alone. But it was surprisingly small. In fact, performance was much better when people moved to the other viewing window and the table stayed still. People are very good at updating where objects will be around them as a consequence of their own motion. So we store visual representations or snapshots, and we also update the locations of objects around us as we move. However, in this experiment, the lab was visible behind the table, so we wondered if object locations are also encoded relative to cues in the room. To investigate this with Hugo Spears, we made an experiment where a cue card could move, as well as the table and the subject's viewpoint. We found positive effects of consistency between encoding and test for object location representations that were either visual snapshots, or updated by self-motion, or encoded relative to the cue card. These three simple main effects, with no interaction, indicate the presence in parallel of all three kinds of representation. Visual representations are egocentric, encoding where things are to the left or right or straight ahead, whereas representations of object locations relative to other cues in the room are allocentric, and either kind of representation could be updated by self-motion. By recording from freely moving rodents, we can see the neural representations underlying our sense of location and orientation directly. This video shows a cartoon of recording from a single neuron in the hippocampus of a rat foraging for scattered food. Red dots show the rat's location when the neuron fires an action potential. Notice that it fires whenever the rat goes into a specific place within the box, independent of its orientation. John O'Keefe discovered these cells at UCL in the 1970s and called them place cells. Each one fires when the rat goes into a different place, and together they tell the rest of the brain where the rat is as it moves around. The complementary representation of heading direction, irrespective of location, was discovered by Jim Rank in New York in the 1980s and has been followed up by Jeff Taubey and colleagues thereafter. We can say something about the environmental inputs to place cells by manipulating the shape and size of the box. The place cells shown here on the left, likes to fire at a specific set of distances from the walls of the box, quite close to the walls to the north and to the west, and a bit further from the wall to the east. These inputs all coincide in the small box shown on the left, but in the larger box on the right, the east and west inputs don't coincide, causing the firing field to stretch out. So with Tom Hartley, we guessed that boundary vector cells that fired whenever a boundary was at a specific distance and allocentric direction from the rat might be the input to place cells. If several of these cells input to a place cell, they would make it fire whenever their firing fields coincide. Like these two boundary vector cells tuned to respond to boundaries nearby to the north and to the west would make a place cell fire in the northwest corner of the box. With Colin Lever, we discovered these predicted boundary vector cells. The firing patterns for one of them in different environments is shown on the right. The boundary vector cell fires whenever there's a boundary nearby to the south, whether it's the walls of the box or a barrier put into the box, or if the rat is put onto two tabletops, shown in blue, pushed together, the cell will still fire whenever there's a drop at the edge of the table close by to the south. It will even fire if there's the gap between the tables nearby to the south. So these cells just respond to the presence of environmental boundaries. More recently, similar object vector cells have been discovered by the Nearim and Moser groups. These fire specifically when there's a small object at a certain distance or allocentric direction from the rat. The firing of place and boundary cells and object vector cells is allocentric in terms of encoding environmental location independent of the orientation of the rat. But we know that perception and imagery are encoded egocentrically, encoding what is left or right or straight ahead of you, corresponding to eye or head-centered neural representations found in parietal cortex. So if we have egocentric representations of boundaries and objects in parietal cortex, shown on the left, and allocentric ones in the medial temporal lobe, shown on the right, we need to be able to translate between them. This requires your head direction. For example, left corresponds to west if you're facing north, but east if you're facing south. This translation process can be done by boundary vector cells or object vector cells whose firing is modulated by head direction. These are shown in the center. Cells whose firing changes with head direction as well as with the presence of objects or boundaries, as you can see here. So with Sue Becker and Andrzej Bikansky, we made a neural level model to explain how egocentric sensory input in parietal areas is translated into allocentric information via gain field neurons in retrosplenial cortex for storage in the medial temporal lobe. The model explains how recurrent connections in the medial temporal lobe reconstruct a coherent scene consistent with a single viewpoint given by place cell firing and a single viewing direction given by head direction cell firing, and how gain field neurons in retrosplenial cortex can then translate this retrieval back into a coherent egocentric scene for imagery. This model predicts egocentric and allocentric boundary and object vector cells and trace and head direction modulated versions of them. And these different kinds of cells are all now being found by Hasselmo and Moser and Lever groups, among others. Now I'm going to talk about another aspect of spatial representation that I mentioned earlier, updating by self-motion. Grid cells in entorhinal cortex, just next to the hippocampus, fire whenever the rat enters one of several locations arranged across the environment in a triangular grid. This cartoon shows the firing of a single grid cell as a rat forages for food, at increasingly unrealistic speeds. These cells were discovered by the Mosers, and neighbouring grid cells have similar but offset spatial firing patterns, like the three shown in green, red and blue on the right. Because these firing patterns have so much intrinsic structure, irrespective of the environment, we think they're primarily driven by self-motion. Usually, self-motion and movement of the sensory environment are locked together. One is a consequence of the other. So it's hard to tell which is driving a neuron to fire. But virtual reality allows us a way to dissociate them. In this task, which I designed with Guifeng Chen, John King, Yi Lu and Francesca Kikuchi, mice run on an air-suspended ball to navigate to stripy poles in a virtual environment where they receive a drop of milk. Every fourth trial, the mouse has to run to a remembered location where the pole has become invisible, which is marked by a cross in the overhead view. Here we can change the gain between the physical environment, the physical movement on the ball, and the visual movement of the virtual world. And this allows us to see whether firing patterns are more influenced by physical movement or visual inputs. The answer is, when simultaneously recording place cells and grid cells, the place cell firing patterns are more influenced by the visual inputs, and grid cells are more influenced by self-motion. If we want to estimate self-location, we must combine environmental and self-motion information, and we think this happens in the interaction between place cells and grid cells. Thus, place cells can be updated by self-motion from grid cells when environmental inputs are scarce, and grid cells can reliably fire in the same location on separate visits to an environment by association to environmental information via place cells. In addition to updating spatial location by self-motion, we think that the grid cells can also do the reverse. The grid codes for two locations can be used to indicate the movement vector required to navigate between them. So, can we detect grid cells in humans? With Christian Dohler and Caswell Barry, we had people perform a virtual spatial navigation task in the fMRI scanner. The task involves exploring a circular arena and then searching for objects like the yellow duck. Memory is tested by being shown an object and being put back into the arena and having to navigate back to where the object had been found. Because different grid cells have firing patterns that may differ in spatial scale and spatial offset, but all tend to have similar orientations, we reasoned that the fMRI signal would differ when running directions were aligned with the 6 axes of the grids, shown in orange, versus when they're running misaligned to the directions, shown in grey. And this is what we saw in right entorhinal cortex. However, we also saw this same signal that varied with running direction 6 times as you go between 0 and 360 degrees throughout the autobiographical memory system. So while grid cells might support self-motion related updating in spatial navigation, they may also support updating of the viewpoint, determining imagery. So we've taken insights from rodent electrophysiology to form a neural level understanding of human spatial memory. But although place cells are found in human hippocampus, in recordings from patients being treated for epilepsy, many more neurons respond to non-spatial concepts, such as the Jennifer Aniston cell shown here, which responds to the idea of Jennifer Aniston, whether it's prompted by different pictures or by her name. In a brilliant experiment, Constantinescu, O'Reilly and Behrens taught people to be experts in bird silhouettes. Ducks having short legs and short necks, cranes having long legs and long necks, and so on. They then watched bird silhouettes changing shape along trajectories in the space of neck and leg length in an MRI scanner. Using the same analysis method we had used, they found similar patterns of grid-like representations as a function of movement direction in this arbitrary conceptual space of leg and neck length. So it's possible that place and grid cells form a system for much more than spatial memory, in which place cells represent states or concepts in arbitrary problem spaces, and grid cells represent the vectors between them, capturing either transition statistics or relational knowledge, and allowing navigation within these conceptual spaces. This is a topic of current work with Tim Behrens and Changmin Yu. Thank you for listening, and thank you for the William James Fellow Award, and thanks again to all my colleagues and collaborators who have made this work possible.